Okay, here we're going to look at initiating an immune response. And while we do have our innate immunity and our adaptive immunity, we're going to focus a little bit more on the adaptive immunity. Keep in mind, our innate immunity is what we're born with, and it includes surface defenses, physical barriers such as our skin and mucous membranes. We're going to look a little bit more about the activation of T and B cells. So the first part is the body needs to be able to identify cells as friend or as foe. It does that with self-MHC markers located on the cell surface. In this case, these markers indicate self and indicate to the immune system that these cells were naturally made and are part of the body. However, in this case, we notice this is a non-self or a foe marker. These antigens are different or foreign. They don't match the host cell markers. As a result, this would initiate an immune response. Macrophages are the ones responsible for inspecting the surface of cells looking for self-MHC proteins. If a cell displays the MHC protein antigen self-non-self combination, the macrophage will secrete the protein interleukin-1 and send an alarm signal to initiate other cells for this immune response to occur. Now, of our foreign body, and our macrophage recognizes that as a foreign body, the CD4 protein is required to authenticate this transaction. The interleukin-1 stimulates helper T cells to initiate two things. On one end, it's initiating a cellular response, which is responsible by the T cells, and a humoral response, which is responsible by the B cells that we see here. Now, the T cells in the cellular response, once activated, the helper T cells secrete interleukin-2, which activates cytotoxic T cells to recognize and destroy cells with the specific antigen found in the antigen-presenting cell. Keep in mind this interaction, the cell-to-cell communication and binding is very important. We have our MHC2, here's our antigen, and here's our T cell receptor. This is activating the helper T cells. This is then initiating the cytokines to be released, and this activated cytotoxic T cell is going through and lysing this infected cell here through this binding that occurs. Now, this method that it chooses to use is very similar to those of natural killer cells, and this stimulates the proliferation and activation of these cytotoxic T cells to help initiate this cellular response, to help control these infected cells that might be circulating throughout the body. Our T cells in the cellular response, well, cytotoxic T cells will also attack transplant tissue and cause graft rejection. So when someone gets an organ transplant, it's the cytotoxic T cells that may cause that organ to be rejected. The drug cyclosporine inactivates cytotoxic T cells. This will be given after a transplant to help reduce the chance of someone's body rejecting the organ that they just received. Now, B cells are responsible for the humoral response. They have antigens here and antibodies is the key part what they produce. So B cells recognize invading microbes but do not go on the attack themselves. B cells are responsible for producing antibodies that inactivate and mark pathogens for destruction by non-specific immune defenses. So here we see a B cell producing a lot of antibodies. These antibodies will be very specific ends here to bind to very specific antigens. Now, the B cells in the humoral response, a little bit more complicated here, but we do see our B cells becoming activated and then producing antibodies, marking this for destruction. Now, B cells can bind free and unprocessed antigens. These antigens are endocytized, processed, and presented on the surface with MHC proteins. Keep in mind, there's MHC markers there. Antigen-MHC complex is indicating, hey, this is an antigen that we need to be able to recognize and be able to destroy because we've now marked this as a foreign invader. The helper T cells recognize this complex, and they will stimulate B cells to proliferate into memory cells and plasma cells, which are the plasma cells which are responsible for the production of the antibodies. And here's our memory B cell for long-term memory. Lastly, just a couple last notes here about the antibody action. All antibodies form an antigen-antibody immune complex. Every antibody has a very specific binding site that matches a specific antigen. Please keep in mind that antibodies themselves do not destroy the antigen. Antibodies inactivate and tag it for destruction. That's their sole purpose. It's this tagging and labeling component. The antibodies themselves are not doing the destruction. They are merely being able to inactivate it and tag it for destruction so that other cells can come about and do the work of the destruction of the pathogen. It's not specifically the antibodies. Antibodies are very important for the binding and identification, but not for the step of actually lysing or destroying the cell.